Hello, everyone. Thank you so much for joining my talk today. My name is Sarah Shapiro, and I am a senior engineer at Mammoth Biosciences. And today I'm excited to tell you some of the work we've been doing on detecting SARS-CoV-2 with CRISPR. The Mammoth mission is to improve lives by reading and writing the code of life. Mammoth was founded in 2018 with the goal of advancing the CRISPR technology into products that can reshape detection and treatment. And we use both a discovery pipeline for new enzymes as well as some licenses from UC Berkeley in order to develop the next generation CRISPR platform that can enable life-saving diagnostics and therapeutics. So all of this is based on leveraging CRISPR's potential, which at its heart is a search engine. And so CRISPR has the ability to be programmed by coupling a Cas protein with a guide RNA that targets a protein of interest. And by modifying the guide RNA, you can target a sequence of interest, whether that be for a virus such as SARS-CoV-2 or for a genetic condition. And then furthermore, these proteins themselves can be programmed in order to perform a targeted function. And so depending on the protein, that could be to read a nucleic acid sequence or to write if you want to edit a certain sequence, or even to knock out certain genes or to modulate them. And so in order to leverage these proteins for developing new technologies, Mammoth has developed a proprietary approach to protein discovery. And so we look at various different sources in order to discover new proteins of interest. And these are fed to a bioinformatics and machine learning platform, which identifies promising candidates. And then those candidates are put into high-throughput experimental screening in order to further narrow down and identify candidates that are promising and can be used in a wide variety of applications, all the way from diagnostics and therapeutics to biomanufacturing and agriculture. So specifically today, I'm going to be talking to you about the diagnostics aspect. And the proteins that Mammoth uses are ideal for diagnostics due to a few key features. One of them is the programmable nature of the platform, where by modifying the guide RNA associated with the Cas protein, you can target any nucleic acid of interest. Furthermore, these proteins exhibit rapid enzymatic recognition, and so they very quickly are able to find their target and convert that into a signal, which can lead to rapid diagnostic assays. Additionally, due to the requirement for the guide RNA to hybridize to the target, there is a high molecular accuracy. And so all of this can lead to very simple and specific assays that can be used for diagnostics. And so for diagnostics in particular, the way that many of these proteins work is that once the protein is programmed by the guide RNA to target a specific sequence, once the guide RNA hybridizes to that sequence and recognizes it, then the complex is activated. And that activation then unleashes nonspecific nuclease activity, where the enzyme begins to nonspecifically cleave other nucleic acids that happen to be nearby. This can be leveraged in order to perform diagnostic applications because you can use a reporter molecule that you introduce into the solution, and that has a nucleic acid sequence that you desire to be cleaved in order to produce a signal. So for example, you can have a fluorophore molecule that's separated from a quenching molecule by a nucleic acid sequence. And so when the sequence is cleaved in the presence of target, that fluorophore is released from its quencher, enabling the solution to become fluorescent. So here is just a video. So the Cas protein is complexed with its guide RNA, and that complex is then able to travel through solution, and the guide RNA programs the complex to bind to its target of interest. And after binding and recognition, the complex is then activated and it begins to non-specifically cleave any other nucleic acids that happen to be nearby. In this case, this is again demonstrated using a fluorophore quencher model where you have a fluorophore separated by a nucleic acid from a quencher, and upon cleavage, that fluorophore is released and becomes fluorescent, allowing you to measure the fluorescence of the solution to determine whether the target was present. And so this CRISPR detector platform can be programmed to target any DNA or RNA sequence of interest. And that can be all the way from an infectious pathogen to a cancer mutation. So simply by changing the guide RNA that is associated with the protein, it gives a great flexibility as far as diagnostic targets. These targets could be anything from viruses or bacteria, or even genotyping if you are interested in identifying a gene mutation or looking at host response. And so these detector CRISPR-Cas platform has a few key advantages that are useful for diagnostic assays looking for nucleic acids. So one of these is that because the Cas protein is interrogating the complementarity between the guide RNA and the target RNA, there's increased specificity when you compare to a traditional nucleic acid amplification test. For example, if you're looking at TACMAN hybridization probes for comparison. Furthermore, these Cas proteins have a native signal amplification, and the protein performs multiple cleavage events, turning over multiple reporter molecules per identification of a single target. And so this can result in robust signal generation from small amounts of amplicon. Additionally, because of the specificity of the Cas protein, it can sort through messy isothermal amplification products. Isothermal amplification tends to be a bit messy, and oftentimes it will produce some non-specific products in addition to the specific product you're looking for. But when coupled with a Cas enzyme, the Cas enzyme can lead to a simplified and faster workflow and fewer false positives due to its specificity. And finally, because the signal generation is just dependent on nucleic acid cleavage, the reporter can be modified to enable a variety of different readout modalities. And so you're not limited to just fluorescence, but you can have very flexible formats for deploying your diagnostic assay. And so as I mentioned, these detector enzymes can lead to increased signal to noise when you compare to just doing an isothermal amplification alone. And so on the left are some example kinetic curves looking at just isothermal amplification, and you can see that it's really hard to tell a difference between the different wells, and all of the curves look fairly similar. But then when you couple this with CRISPR detection, as shown on the right, the specificity of the CRISPR-Cas complex leads to good differentiation in between your positive and negative wells. And in this case, the positive wells are shown in orange and the negative in red, and you can see there's a very distinct signal difference in between the two. Furthermore, the specificity of this detector system can enable detection even down to single nucleotide differences, if you're looking, for example, at single nucleotide polymorphism. And so the image on the top illustrates this by showing a target that has been mutated in various locations, and the target that was designed, that the guide RNA was designed for, is shown by the boxes in pink, whereas there was a comparison to different point mutations mutating to the possible different nucleotides at all the different locations. And we see that the CRISPR-Cas complex has really good specificity identifying its target, and it's resistant to a single nucleotide change all the way from the region right next to the PAM down to the very end of the spacer region. And so we can leverage this at Mammoth in order to be able to genotype and identify differences between a full wild-type gene, a heterozygous, or a full mutant. And we can compare the signal and very easily stratify into different buckets and allow us to determine for the samples shown along the bottom whether they were a wild-type, heterozygous, or a mutant. Additionally, because the detector platform is compatible with different readout modalities, it can enable application to different types of diagnostic technologies. And so I talked a bit about fluorescence previously as a readout, but the assay can also be coupled to a lateral flow readout, where you have, in response to the cleavage event of the CRISPR-Cas complex, you generate a different signal on a lateral flow strip. And so you can observe this change by eye, similar to the way you would, for example, in a home pregnancy test. Additionally, the CRISPR-Cas complex can be coupled with a secondary enzyme that will generate a colorimetric change. And so in this case, you can have a change in color in response to the presence of target. And that change in color can be identified by eye. And a final option is to do electrochemical-based detection using instead a reporter that is electrochemically active and exhibits a different electrochemical signal after cleavage. All of these different advantages of CRISPR enables it to have broad application across the full continuum of testing, all the way from laboratory tests to at-home tests. And the programmable nature of the detector platform means that you can use it to detect any different indication, while its high specificity and sensitivity provides great usefulness in very different diagnostic settings. And so this can be useful both on a laboratory basis, where the goal is to increase throughput and using, for example, automated tests in order to run as many tests as possible, all the way from that to at-home tests, where you're interested in having a single-use disposable device where someone could test at their own home. And in order to do this, you would need something that is low cost and very rapid, so the person doesn't have to wait a long time when running the test, as well as be able to be distributed into homes. And so the current COVID-19 pandemic has really validated the need for rapid and accurate diagnostic tests that can be quickly deployed. And Mammoth worked very rapidly in response to the beginning of the pandemic. Mammoth first developed an assay very rapidly in response to the publication of the full viral sequence in January of 2020. And we worked with clinical collaborators at UCSF in order to develop first a peer-reviewed manuscript that was published in Nature Biotechnology in April of 2020. We further were able to obtain partnerships with GSK and with DARPA to develop decentralized testing options, as well as some funding from the NIH in partnerships with MilliporeSigma, Hamilton, and Agilent in order to develop a lab-based test called DETECTRT. And all of the efforts surrounding the assay has enabled us to go from paper to accelerated product development in under 6 months. I'm going to talk to you a little bit about both the lab-based tests as well as decentralized testing. But first, I would like to give an overview of the assay itself, which is published in a manuscript in Biotechnology in April of 2020. And this was the world's first peer-reviewed manuscript demonstrating the use of CRISPR diagnostics for COVID-19. And so in response to the pandemic, Mammoth scientists worked very quickly in order to develop an assay that could leverage CRISPR and use it to detect SARS-CoV-2. And so we developed two different assays for two different pieces of the viral genome. One is the E gene region, where the guide RNA that we developed overlaps with the WHO E amplicon, and also an assay for the N gene region, where the guide RNA overlaps with the CDC&2 amplicon. And so the first step in developing the assay was to make sure that the guide RNAs were specific for the target of interest and didn't have cross-reactivity. And so the graph in the bottom left shows a comparison of the guides for the N gene and E gene target, as well as RNase-P, which we use as a sample input control. And we see that the N gene and the E gene guides are both very specific for that particular target and don't show cross-reactivity with some other common coronaviruses, whereas the RNase-P, or the sample input control, is present across the different coronavirus samples. Furthermore, Mammoth leveraged a lateral flow-based readout in this test in order to eliminate the need for bulky fluorescence equipment. And this lateral flow test will determine, based on a visual readout, the result of the assay. And so as the Cas complex is activated, it cleaves the reporter that when the product of the reaction is put on a lateral flow strip, if the reporter has been cleaved, then it generates a different signal compared to if the target was not present and the reporter was not cleaved. And so the CRISPR portion of the assay works incredibly rapidly. And so the graph near the bottom right shows the signal after different reaction times of the CRISPR part of the reaction. And we see that almost instantaneously, you can see a difference in between the positive and negative samples. The lateral flow readout is shown in the bottom right, and we see that we can start to distinguish at about 2.5 minutes, but after 5 minutes, you really see a strong difference in between your positive and negative samples. Mammoth further validated the test by collaborating with UCSF and testing on real clinical samples. And so the slide shows a comparison from tests with 83 different clinical samples. And so if both the N gene and E gene assays were positive for SARS-CoV-2, then the sample was called positive. If either one of the N or N gene or E gene were positive, then the sample was presumptive positive, whereas if both the N gene and E gene were negative and the RNase-P control as a sample input control was positive, then the sample was called SARS-CoV-2 negative. And using that scheme, we were able to obtain a positive percent agreement of 95% and a negative percent agreement of 100%, illustrating the applicability of CRISPR to real clinical samples in response to the emerging pandemic. And so Mammoth further leveraged the assay in order to develop a lab-based test that could impact high-throughput testing that's done in diagnostic laboratories. So that is the DETECTR-Boost SARS-CoV-2 reagent kit. This is in partnership with Agilent and their Bravo instrument, as well as Hamilton and their STAR instrument. And it has the potential of increasing testing capacity up to thousands of tests per day due to the simple and rapid CRISPR test. And as a result of this, can get results faster, up to 2 times faster than PCR workflows. Furthermore, because the assay is very simple, there is less hands-on time. It's less than an hour, which reduces the FTE load and can allow employees to focus on other tests while increasing diagnostic throughput. And finally, it has a performance similar to RT-PCR with an LOD of 150 copies per milliliter. So in comparison to RT-PCR, the DETECTR-Boost assay takes approximately 90 minutes per 96-well plate in order to run, whereas RT-PCR takes about 140 minutes per 96-well plate and also involves some manual steps. For the DETECTR-Boost assay, we have a duplexed assay with SARS-CoV-2 N gene as well as RNA-seq as an internal control, and it displays an analytical LOD at 150 copies per milliliter. Furthermore, in a clinical study with 230 nasopharyngeal swabs, we were able to obtain 100% positive percent agreement in samples that had a comparator CT that was less than our estimated LOD. And so in addition to applying the CRISPR detector platform to laboratory-based testing, Mammoth has also worked to develop a decentralized testing platform. And so for the assay on the decentralized prototype, it's able to perform the full assay in less than 40 minutes, including extraction, amplification, and the detector CRISPR reaction. And so to develop this prototype, we integrated the assay chemistry onto a prototype device that only requires a single user step and allows for sample to answer in just 40 minutes. Mammoth tested this on 100 different prototypes and found that we were able to obtain 96% overall agreement for detection of SARS-CoV-2. And we've manufactured more than 500 of these prototypes. Furthermore, these prototypes also convert the signal from the assay into a positive-negative indicator light in order for the results to be unambiguous to the user. And so at the end of the assay, there is a measurement cycle that is initiated, and then the data is collected, and an algorithm calls the result, and then based on that result, the appropriate light is lit up, whether that's a positive or a negative call. And as I mentioned, we tested this on 100 different prototype devices and found that we were able to obtain a positive agreement of 100% and a negative agreement of 92% with overall agreement of 96%. And so all of this illustrates the applicability of CRISPR and the advantages of having the highly specific and rapid testing that can really impact the future of diagnostics and can enable molecular testing that is in the palm of your hand. And with that, I'd like to thank the entire Mammoth Biosciences team. Mammoth has a lot of great people who work here, and it's a pleasure to come to work with them every day. As well as I'd like to give a shout out to all of our collaborators, including UCSF for the Nature Biotech paper and the clinical studies, as well as the many other collaborators on the various projects we do at Mammoth. Thank you so much for listening to my talk.